Hello.
Hello, sir. Good morning, sir.
Sir, good morning, sir.
Hello. Can you hear me?
Yes, sir.
Komal,
Yes,
Komal,
sir. Yes, sir.
it's okay.
Yes, sir. We'll start. The beautiful thing about learning is that nobody can take it away from you. B.B. King. With this beautiful quote, a warm welcome to respected speaker, principal sir, staff, students, and participants to this webinar. Faculty of Pharmacy, Dr. MGR Educational Research Institute is successfully organizing series of national and international webinars and FTP with eminent speakers from all around the globe with a vision to intensify the intellectual wealth to the students, faculty, research scholar, industrialists, and pharmacists. In continuation with the events conducted, Faculty of Pharmacy, Dr. MGR Educational Research Institute is organizing this national webinar, which is to emphasize the pivotal role and need of current drug discovery on the topic Overview of Drug Receptor Interactions on May 31st, 2021 by Dr. Biju Mathur, Associate Professor, Department of Pharmaceutical Chemistry and Amrita School of Pharmacy, Kochi, India. Accurate identification of potential interactions between drugs and protein target is a critical step to accelerate drug discovery. Despite many relative experimental researches being done in the past decades, detecting drug target interactions remains to be extremely resource intensive. So let's gain knowledge in this extremely interesting topic of drug receptor interactions. A recorded version of this webinar will be available in our YouTube channel named Principal Pharmacy. You can subscribe to this channel for regular updates of webinars and information. Now let's move on to the session. May I now request our beloved principal, Dr. N. Harikrishnan sir, who is a pillar of support and encouragement, to deliver the welcome address. Over to you, sir.
Thank you, Mrs. Komal. Thank you. Thank you very much for the highlights of national webinar. So here, today, respected dignitaries and below the faculty members, students and participants, good morning to all. So as you know, the Faculty of Pharmacy, in continuation with its successful online webinar and mainly the public awareness activities, is now organizing this online awareness program with renowned experts from different fields, especially for pharmaceutical field, with an aim bring more awareness of COVID-19 and COVID-19 as well as the role of drug discovery with the COVID-19 during the pandemic situations. So at this moment, I would like to extend my heartfelt gratitude to our founder, Chancellor Dr. A.C. Sanmugam Sir, Honorable President, Engineer through A.C. Arun Kumar Sir, and our Honorable Secretary Group, Ravi Kumar Sir, under whose benevolence all our efforts are transforming successful realization. I take the opportunity to welcome here the today speaker, Dr. Biju Matthew. So Amrita School of Pharmacy, Kochi. So he is going to present overview of drug receptor interactions. So overview of drug interaction receptor. Why this topic is very important for being a pharmacy profession. Nowadays, the SARS-CoV-2 has been marked as highly pathogenic coronavirus of COVID-19, a disease in the human population, causing over 5.5 million confirmed cases worldwide as COVID-19 has posed a global threat with significant human casualties and severe economic losses. So there is a pressing demand to further understand the current situation and develop rational strategies to contain the drastic spread of the virus. Although there are no specific antiviral therapies that have proven effective in randomized clinical trials, currently the rapid detection and technology, along with several promising therapeutics for COVID-19, have magnified its drastic transmission. But though mainly here, the drug discovery being a pharmaceutical profession, they involved to find the various research molecules towards the COVID-19. So recently, even Dr. Reddy's laboratories also, they introduced 2G, and as well as many companies, they are come forwarding to launch new drugs. So here, I would like to thank, once again, I welcome all participants to this webinar, national webinar, this main topic, 2021, and wish everyone to great learning experience. So thank you for joining us. Thank you very much for. So please, those who are participated here, so please mute your mics, and if you any doubts, so finally, so after the session, please post your questions towards the chat box. Thank you very much, Komal. Thank you.
Thank you, sir. Thank you for your welcome speech. May I now request Mrs. Gajlakshmi ma'am, Associate Professor, Faculty of Pharmacy, Dr. MGR Educational Research Institute, to introduce the speaker of today's webinar. Over to you, ma'am.
Thank you, Mrs. Komal ma'am. A very good morning to one and all. I am profoundly delighted to take an opportunity to introduce the speaker for today, who is going to deliver a presentation on topic and overview of drug receptor interactions. Dr. Bijo Matthew sir is Associate Professor of Pharmaceutical Chemistry at Amrita School of Pharmacy, Cochin. He obtained his MPharm in Pharmaceutical Chemistry from Tamil Nadu Dr. MGR Medical University in 2008. He has completed his Pharmaceutical Science from Jawaharlal Nehru Technology University, Hyderabad in 2016. His primary area of focus are in drug discovery of neurodegenerative disorders, cancer, and molecular modeling. His main research interest is on the design of enzyme inhibitors, especially monoamine oxidase and acetylcholine esterase inhibitors. Sir has published more than 145 papers in national and international journals with H index around 27 in Scopus. Sir has totally 13 years of teaching and research experience. He has authored one book, edited one book, and he has published 5 book chapters. He has been invited as invited speaker in national and international level seminars around 25. He is serving as a chief executive guest editor of the journals of molecules, current topics in medicinal chemistry, current drug targets, and combinatorial chemistry, and high throughput screening. He is a section editor of the Journal of Coronavirus under the publication of Bentham Sciences. Sir has received Amrita Vishwa Vidyapeetham Chancellor's Publication Award in 2021. He received 1 patent from Korean Intellectual Property of Office in the topic of protective or therapeutic composition of neurological disorders containing furanochalcones as effective agents. So without consuming any time, I would like to invite a speaker for the forum with a small positive invoking. Knowledge becomes power only when we put it to use. We welcome you, sir.
Thank you, ma'am. May I now request today's speaker to start the session. Over to you, sir. Participants are requested to mute their audio and not to present during the talk. Over to you, sir.
Yes, can you hear my voice clearly?
Yes, sir.
Can you see my slides?
Yes, sir. Yes, sir. It's visible, sir.
What about the audience? Is it clear, my voice?
Yes, sir.
Loud and clear, sir.
Hello, hello.
Clear, sir.
Yes, yes. Okay, today it's a very familiar topic to all the pharmacy profession, overview of a drug receptor interaction. Hello?
Yes, sir.
Yes, sir.
Yeah. Okay. So the main objective of this particular seminar, so we are just seeing the basic concepts of drug receptor interaction, especially connecting with the forces between the drug and receptor, the various types of forces, and how these forces is able to interact with the receptor in an effective manner. So that is the basic concept only we are going to see that. Okay, we'll see the first slide. Can you see the slide clearly?
Yes, sir. It is visible.
Okay. So you see here. So today we are going to deal with only the drug and receptor. So there is only the two terms. So we need a drug molecule and a receptor. So this is a very famous German scientist. So who really established the basic concept of drug receptor theory in the 18th century. Okay, so his name is very common to you. I think he was a very Paul Enrich. So he only established a particular theory which is connecting with the matter and the biological target, which is able to get a biological response once it will interact. So it's very famous theory. Can you see the animations clearly?
Yes, sir. The animations are good and clear.
Yeah, yes. This is his famous theory. An agents, it is not act unless they are bound. So today we are going to analyze what are the bounding type of techniques. So especially connecting with an organic molecule and a biological receptor. So this is a very simple concept. This is his very famous concept. The agents not act unless they are bound. Okay, so once it will bound, you can see a drug receptor complex. After that, you can see a series of biological response. So that biological response, obviously, it will benefit to the patient, so it will be helpful for your drug design. So this is his very famous theory. So can I go to the next slide? This is a basic introduction of the introduction of drug receptor. Okay, so can you see these slides clearly? Also, this is a very famous concept of drug. You know that drugs are, you see here, can you see the slides? Hello?
Yes, sir. Yes, sir. It is
So
visible.
this is a very simple concept. So drugs can be either stimulatory or inhibitory. This is a very famous concept. So there are more than 25,000 of drug receptor targets in our body. So many drug receptor targets are nowadays is available in your protein data bank. So this is a basic concept. Drugs can be stimulatory or inhibitory. And you can see the basic concept already you studied in your pharmacology. So this is a common concept of the classification of ligands. So that is the classification. I can classify by means of agonist, partial agonist, and antagonist. So I think I just mentioned its specialty by means of affinity and efficacy. If you are seeing for the affinity of your agonist, so you can see that affinity and efficacy, they are 100% in manner. But if you see in the case of your partial agonist, you can see the affinity, but efficacy. So maybe you can see a degree in the efficacy. But if you see in the case of your antagonist, you can see affinity, but at the same time, the lack of affinity. So this is a simple concept of the affinity and efficacy, which is connecting with your various types of ligands. Clear? Hello?
Yes, sir. Clear, sir.
Yeah. So this is
Yes, sir. Yes, sir. Yes, sir.
yeah, this is a very simple concept. I am going to establish the various types of drug receptor interaction. So before that, I'm just establishing what is a specialty of a drug molecule. You know that, so if you are going to synthesize a particular molecule, so you should keep some basic criteria that should satisfy according to a property. That property I can name it as it is drug likeness. So I will tell you some of the basic property which is able to make your molecule as a drug form. You know that there are lakhs of molecules I can synthesize it in my lab. Unfortunately, very few of them, I think approximately, if you are concerning about the FDA approved drugs, I think approximately 2,500 or 2,600 only FDA approved drugs you can see which can be considered as a drug molecule. Okay, so if you want to design your drug, so I will introduce some basic property. It should not be more molecular weight, more than 500. It should not be more lipophilic. It should not be more hydrophilic. And you can see some the functional group, which is the crucial feature which can able to interact your receptor. So that is a basic concept. That is a specialty of your drug molecule. And we will see, sorry. Okay, so this is your target. So target, obviously, we know that that is the area you can see in our body. Obviously, it will lead to the biological response. And once your drug molecule interacts with your receptor, and you cannot expect any type of toxicity. So that is a main aspect of drug design concept. So once it will interact with the receptor, so you cannot expect any type of toxicity. So you need to design a molecule with a high efficacy and a toxicity free. So that is a basic concept. Okay. Okay, this is the, I think commonly you know this concept drug. Once it will interact with your receptor, you will get a drug receptor complex. So that complex primarily will able to produce a biological response. You know that it is maybe reversible and irreversible type of interaction. You can see that. So that is typically depends upon the nature of your drug molecule and your target. At the same time, your therapeutic profile depends upon your nature. You can see that whether it may be reversible or irreversible. So we are going to see only the basic concept. So not going to deal to the design of your molecule, how it will interact. We will see that what are the types of interaction is commonly you can see for a drug receptor interaction. Can you able to follow clearly?
Yes, sir.
Yes. Can I go to the next slide?
Yes, sir.
Yes. Okay. So this is a very simple concept. A drug. Can you see the animation picture clearly? So whenever if you are designing, so if you are making your drug molecule, there may be a global energy is situated or can be present in every molecule. So once it will approaches towards your receptor, the molecule will try to minimize its energy for getting a maximum interaction. So that minimization, I can simply tell a term, it is bioactive conformation. So that is a busty conformation of your molecule, which is able to react to your molecule in an efficient manner. That means it is able to interact with your receptor with high stability. At the same time, hello, please mute your audio. Hello. Hello.
Yes,
Hello.
muted, sir.
Yeah. Let me go to the next slide. Okay, these are the major types of interaction. So I will discuss today. So I think commonly every people will know the types of bonding only. So covalent type of bonding, ionic, it is dipole interaction, hydrogen bonding, charge transfer interaction, hydrophobic interaction. Wonderful. There are some other forces are there, something like pi cation, London forces, many bonding types or interpretations available, but these are the basic 7 types of bonding, which we are going to see that how these types of bonding will efficiently interact with your receptor. Okay. Can I see the next slide? The first we are going to see the covalent type of bonding. Can you see that? The next slide, is it visible?
Yes.
Yeah.
Yes, visible, sir.
Yeah, this is a very famous concept. Can you see my drug molecule, which I mentioned in yellow? Can you see that?
Yes, sir.
Yeah, so this is the most, you see here, whenever I am using these slides, I will only demonstrate by means of animations. Obviously, it will give, what is that, more idea. So what are the basic concepts? If you are seeing this particular drug molecule, so I can see that I just mentioned one part. Can you see my cursor clearly? I just mentioned some of the area in green. Can you see that?
Yes, yes, sir.
Yeah. So this is the area. I can tell that it is a warhead. This is the primary functional group, which is able to interact with your receptor by means of in a irreversible manner. You know that covalent bonding in an organic perspective of view, covalent bonding is nothing but it is a mutual sharing between 2 electrons. So that 2 electrons ultimately will generate the formation of a bond. Obviously, that bond formation will ultimately will give the strength of your molecule. That means it will give the bond length of your molecule. That is a basic concept, you know, in organic perspective of view. So this is a basic concept. I can see that in a covalent bond inhibitor, you have a prominent function group. I can tell that it is a warhead. That is the particular area which is going to interact with your receptor. Okay. Can you see that next animation? Hello?
Yes, sir. Yes, yes.
If you are
Yes,
seeing
sir.
this particular animation, you can see a clear factor. Your drug is able to interact with the receptor. Fortunately, if you are pointing out that green part, that is a major area which will bind to a receptor. So there are many other areas in your drug molecule, some functional groups, some hydrophobic phases, some what is that, pi electrons, many things are there. So fortunately, the only one area which can make a covalent type of bonding, so that bonding or that functional group, I can tell you that it is a warhead. Okay, it is a irreversible type of covalent bond. So I will demonstrate a very simple example. I think in your medicinal chemistry, for a B-farm case, it is very familiar for the aspirin mechanism of action, especially. So if you are concerning about the drug design of COX, so you can see a clear factor. You know that aspirin, basically, it is an acetyl salicylic acid. So can you see the interaction clearly? Yeah, so this particular interaction, so you have the structure of benzene, you have the structure of acetyl group. Once it will interact with your physiological pH, your COH will be ionized. And if you are seeing the case of your acetyl group, that is the only warhead. It will interact with your serine 530. So there are some other residues, something like tyrosine and something like guanidine residue. So that may be useful to enter your hydrophobic cavity of that particular region. So only the acetyl group will make the acylation of your serine residue. That is a typical residue which will able to acylation of your aspirin. That acylated form, it will ultimately will make the prostaglandin synthesis. It will just arrest the prostaglandin synthesis. That is a primary pharmacology. You know that. So I can tell you it is a very strong bond, and it is an irreversible type. Obviously, I told in the initial time. And you can expect a target disruption. You know that once your drug molecule interact with your receptor, so you can see that there is no chance for the formation of de novo type of drug design, that is de novo type of protein synthesis. Obviously, we know that you can expect a target disruption. Okay, is it clear, audience? I think this is very simple concept only. That's why I'm just moving in a very faster manner. Is it able to understand clearly?
Yes, sir.
Yes. Is there any problem? You can type me. Okay. So this is ionic bonding. You know that ionic also, we know that in an organic perspective of view, it is a mutual sharing. Sorry, in covalent, it is a mutual sharing. If you are seeing the case of your, ionic, you can see that it is a one electrostatic force of attraction. Okay, so there may be a transfer and there may be acceptance. Okay, if you are seeing the case of your ionic bonding. Okay, so this is a basic concept. Can you see the animations clearly?
Yes, sir.
So this is a drug molecule with a carboxylic functional group in an ionized format, and your receptor maybe contains an NH3. And I will also make an explanation drug molecule with an amine group and a target with a carboxylic group. So both, you can see that a positive and negative poles are situated between your drug molecule and your target. You see here, whenever if you are introducing a drug molecule, you know that they are neutral molecule. Once it will interact with your receptor, then only you can see what is that a ionized form. That ionized form is able to make an electrostatic force of attraction between your drug and your receptor. So this is a very famous interaction between a pivacabine and your drug molecule between a guanidine receptor, which is present in your molecule. So it is a type of reversible, effective. It is greater distance between the charges. Okay, and it is one of the major thing I told you know that it is very important. Once your drug molecule will ionize only at a in a physiological pH. Can you see the next slide? It is dipole interaction.
Yes, sir.
Yeah, so this is a very simple concept only. So if you have seen the case of a drug molecule, I think my drug molecule functional group is a ketone. So can you see the cursor clearly? I just mentioned ketone. You can see, expect a positive and negative pole between your carbon and oxygen. You know that oxygen is highly electronegative atom. Obviously, it will withdraw the electrons more towards its own side. That will give more electron density, and you can see a negative charge. At the same time, you can expect a deficiency in the side of carbonyl carbon atom. So that deficiency will able to interact with a carboxylate anion, which is present in your binding site. At the same time, your carbonyl oxygen may have a tendency to interact with your amine groups, which is obviously a positive charge at a physiological pH. So you can expect a positive and negative type of interaction between your drug molecule and your receptor, especially if you are concerning about a dipole in your drug molecule. Clear? So this is also a net dipole between a ketone. Okay, I think very similar example. So very many examples. So you can see that. Okay, I will tell you. You see here, these are two polar. Only the two polar molecules will able. Your polar is your one of your molecule, or that means your drug molecule, and the next polar is your receptor. It is very weak. Obviously, we know that it is a weak electrostatic force of attraction. So can you see the next slide clearly?
Yes, sir.
This is hydrogen bonding. I think whenever if you are explaining in molecular docking studies, you know that for docking software, and it's you can see this particular aspect, hydrogen bonding. Okay, so this is one of the simple aspects. I will demonstrate you hydrogen bonding. Can you see that the basic concepts of hydrogen bonding? Can you see that?
Yes, yes, sir.
A carbonyl group between your NH and carbonyl group between your OH and a N, especially a tertiary amine between your NH. Okay, can I use my cursor clearly? Okay, I will. Hello, can you see my pointer option clearly?
Yes, sir. We
Yeah,
are able to.
yeah. Let me tell you that I will tell you a simple demonstration of hydrogen bonding. So if you are seeing that hydrogen bonding in a definition view, it is a simple type of bonding between two electronegative atoms. Clear? So I just mentioned my electronegative atom, that is blue and red. Your red part represent your oxygen and your blue part represent your nitrogen. Clear? Hello?
Yes, sir.
Yeah,
Yes, sir.
it is between the bond between two electronegative atoms. So in that, one is a covalent bond, another is a weak van der Waals force of attraction. So then I will tell you that how this van der Waals force of attraction is able to interact. This particular nitrogen, you know that it is most electronegative. It will withdraw the electrons, and you can expect a high electron density, and you can see electron deficiency in your hydrogen atom. So that hydrogen atom is able to make the next pole, which is having high amount of electron density. So that interaction I just represent by means of an yellow lines. This particular area I can tell you hydrogen bond donor, and this area I can tell you hydrogen bond acceptor. So this is a very famous concept. Whenever if you are designing a particular drug molecule, your hydrogen bond donor and your hydrogen bond acceptor. Okay, clear? Is there any doubt? Can I move to the next animation? Hello.
Yes, sir.
Okay, so this is the explanation. Hydrogen bond donor and hydrogen bond acceptor. Whenever if your molecule is a functional group with a hydrogen with an electronegative atom, it can able to make the hydrogen bonding. That particular group, I can tell you a hydrogen bond donor. At the same time, if your group is able to receive your hydrogen atom by means of a van der Waals force of attraction, that I can tell you it is a hydrogen bond acceptor. Clear? So this is a very famous example I can explain by means of a salicylic acid. This is intramolecular hydrogen bond, and intra, you know that within your molecule, and inter is between that. There is, I think these are the basic concept. Okay. Hello, can you see the next slide? It is charge transfer interaction. Hello?
Yes, sir. Yes, sir.
Okay, it's a charge transfer interaction. Hello?
Yes, it's visible, sir.
Hello? Hello.
Yes, sir.
Yeah.
It's visible.
Can you see the next animation? Oh, sorry. Okay. Can you see the this very famous charge transfer interaction? I just explained my drug is a very famous antifungal molecule. So can you see the specialty of these two molecules, my dear audience? Can you please explain? So what is your observation by seeing these interaction? Hello.
Yes, sir.
Yeah, anyone, any. I think you can act. I think there are many B pharmacy and then pharmacy students are there. These are the basic concept. By seeing this interaction, can you able to interpret your idea? What type of category you can expect between your drug molecule and your receptor? You know that receptor is a tyrosine type of residue because obviously it is a phenolic moiety. So can you please explain or can you please interpret what is your observation? Anyone, please respond. Hello, hello, hello.
Yes, sir.
Yeah, anyone, can you please explain what is your observation by seeing my animation? It's not my animation, it's a animation which I got from the book. Okay, please tell me.
Wi-Fi is taking.
Hello? Yeah, it's a pipe stacking that I know that. You please tell me the specialty of your molecule or the nature of your molecule. That is a thing I'm going to explain.
Migration of the electron from the phenol substituted derivative towards the antifungal derivative.
Yeah, why? Why it is happened? That is a basic concept of organic. Okay, no need to give the answer, especially faculties. The students, can you please tell me your ideas? Then only I can go to the charge transfer interaction. Hello? Any students? Any students? You please tell me your observation. Tell me, tell me. Hello, hello. 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 Hello.
Yes, sir.
So any students, can you tell your observation? What is the basic, the observation? You can see that someone is telling there is a charge transfer is migrating. Can you see that what is the reason behind in that charge transfer? So that is a concept we are going to explore.
Interaction between aromatic ring.
Why that interaction takes place? So I think the concept is very simple. If you are seeing the drug molecule, can you see my pointer option?
Since they contain pi bonds, that's why.
Tell me once again.
Since they contain pi bonds, so pi is taking.
So that is a basic concept only. Obviously, you can see pi electrons, pi stacking will be there. So before that, why the pi stacking is able to from this? Okay, I will take my pointer option. Okay, allow option. Okay. Okay, can you see the pointer option clearly?
Yes, sir.
So you can see that if you are concerning about the groups which is bearing from your benzene, they are highly electronegative. Is it right? Is it right or not?
Yes, sir.
Yes, yes.
So that means if you are concerning about this particular antifungal molecule, you can see that there is a crowding of please mute your audio. Let me explain. Okay, so if you are concerning about the benzene system, you can see that the crowding of electronegative, something like cyanide group, you know that this is a very highly electronegative group and a halogen. So that electron withdrawing capacity around that, surrounding by your molecule is able to make less electron density in this particular area. Clear? Clear or not? Hello?
Yes, sir. Yes.
Yeah. So at the same time, you can see a residue from the receptor. It is an electron donating group which is able to interact with your bonding, sorry, which is able to interact with your lone pairs by means of resonance. So you can see an excess of electron. In both cases, you can see the benzene system, but here the excess of electron rich is able to make a charge transfer towards your adjacent molecule, which is obviously keeping some less degree of electron density. So that is a simple concept. I can tell you that if your molecule have high electron negative groups in your structure, obviously they are behaving a lack of electron density. That lack of electron density is able to receive the electrons from a receptor, especially which have a pi electron richness. Clear? Clear or not? Hello? Hello?
Yes, sir.
Yeah. Okay, now we will see that. Okay, I will tell the demonstration, sorry, conclusion. It is interaction between electron donating group and electron acceptor. This is electron donating, this is acceptor. It is a fraction of electron density. I can see, I can already, some other person who told you that it's a fraction of electron density, something like pi or pi cation, pi anion type of interaction you can expect. This is hydrophobic. Can you see the, I think we are coming to the end of the section very soon. This is a hydrophobic interaction. Okay. Can you see that? I think this particular picture I copied from the, what is that? Richard B. Silverman, Organic Chemistry, Drug Design. A very famous Elsevier book I just copied. Can you see this interaction? I think the picture itself give a clear idea that, okay, what type of observation, what is your mind by seeing the picture? What are the types of observation you can get initially? Hello, hello. Hello.
Yes, sir.
Yes,
Yeah.
sir.
So you can see that your receptor contain a non-polar chain. I think there is no need to explain what is meant by non-polar chain, and your drug molecule contains a non-polar chain. So before the interaction, you can see that your molecules contain surrounded by one, you know that it is happens between a physiological pH, obviously participation of water molecule, that is a common aspect. You can see that your water molecules, they are in a ordered form. Can you see that? Is it a ordered form or disordered? Is it a ordered?
Yes, sir.
Yeah. So once it will interact with your receptor, between your non-polar chain, between the receptor and your drug molecule, so it will be scattered, disordered. That disorderedness will make the interaction. Okay. So I think that particular concept I explained by means of what is entropy. Okay, this is a concept already told you. There is this is an increasing entropy. Therefore, the results a decrease in the free energy, which is stabilized. You know that what is meant by entropy. Okay, this is a very famous, what is that, anti-convulsant drug, which can able to react with your charge, especially hydrophobic interaction. Okay, so your benzene system from your drug and your phenyl system from your receptor. Can you see the next slide clearly?
Yes, sir.
Yes, sir.
This
Yes,
is
sir.
the van der Waals force of attraction. This is a very simple concept only. I think so very negligible kilocalorie is responsible for this particular interaction. So it is between a dipole between a charged molecule and your receptor. Okay, I will give the explanation. Very temporary. It will interact with a nearby dipole. You can see that. So this is the last one I can tell you. A Dibucane, very famous local anesthetic. So if you're seeing the particular drug molecule, you can see various types of features. You can see a quinoline ring. You can see a butoxy. You can see a amide. You can see a tertiary amine. So you can expect many type of interactions once it is able to interact with your receptor, especially hydrogen bond, hydrophobic, and ionic, halogen type of bonding. Many things you can see that. Clear? Can you see the last slide? I two more slides only. Okay. Please be patient. Hello.
Yes, sir.
Yes, sir.
This is a simple demonstration I can explain you. I think you can get these images from the Google. Very simple. You see here concept, agonist, antagonist. You see here both agonist and antagonist. You can expect similar type of function group. Not in all cases, but anyway, you can expect similar type of functional group. But can you see these red mark? Can you see that? Sorry, so it's black. Can you see that?
Yes, sir. Black.
So that is the extra functional group which may be able to with your receptor will make yes inhibitory effect. You know that already in my beginning slide I explained what is meant by stimulatory, what is meant by inhibitory. So in case of your antagonist, that extra functional group will make an inhibitory effect. So ultimately, it will give what is that? Additional interaction. That additional interaction is able to make more affinity with the seer and efficacy. You know that what is meant by in the initial slide itself. I told you that. Okay. I think we are going to see the second last slide, the common features of drug receptor interaction. Okay, we are concluding. This is my simple conclusion. The binding occur, which is a point type of attachment between a functional group and the functional group which is present in your receptor. That is the first point I can tell you. You know that this is a very famous, the concept, the function groups always, that means the presence of function group in your drug molecule, which will just refine or which will shape your molecule by means of electronic and steric type of interaction. So that is the best thing which is able to make your binding process. I can tell you that this is a binding process. Okay, so that binding process will give electronic and steric parameter. Obviously, these two parameters will controlled by your functional groups, which is present in your molecule. Clear? Clear?
Yes, sir.
Yeah. This is a long, the next thing, bonds could be either intermolecular and intramolecular. Sometimes if your molecule is more rotatable bonds, once it will interact with your receptor, you can expect the minimization of energy. That minimization sometimes will able to make the intermolecular. At the same time, you can expect a intramolecular also. Okay. The next is, this is the last concept. If you talk about the reversible binding drug to receptor should be in an equilibrium state. I told you, that. So there may be association constant between a drug and receptor. That may be a dissociation constant once it will, after the interaction with your receptor. That means after the formation of biological response. Clear? This is a conclusion. What is a conclusion? Very simple concept only. The conclusion is the driving force between your drug and receptor is always occurs in very what is that low. I can tell you that it is a low energy state. That low energy state is the best state which may be able to interact with your bioactive conformation of your molecule and your receptor, either inter or intra, various types of forces. I told you already something like reversible or irreversible. Anyway, the driving force is nothing but you need to make your drug molecule with a low energy state. And the second concept is the biological activity. You know that it is typically depends upon the drug affinity between your receptor and your drug, that will ultimately give your stability. And the last concept I already told you, that the dissociation constant, which will give to a best explanation how the drug molecule is potent. The degree of potency, obviously, it will determine the dissociation constant of your molecule. Okay, I know that this is a very simple demonstration. Once you are able to know this basically, then only we will go for the molecular docking or something like of higher types of computational methods for the drug discovery. Clear? Any doubt in this? Can I just find out? Okay. Okay. Tell me, you have any doubt in this?
Thank you, sir. I should say that was a very well constructed presentation, which has made us all have a very good clinch on the receptor interactions. Not only our topic, even your talk was also highly interactive, and these animations made us all have a very clear idea about the drug interactions. So before we move on to the question session, I would like to request our beloved principal, Dr. N. Harikrishnan, to present the e-certificate to Dr. Biju Matthew, Associate Professor, Department of Pharmaceutical Chemistry, Amrita School of Pharmacy, as a token of our gratitude and appreciation towards sharing his valuable knowledge and expertise among us. Principal, sir.
Thank you, Dr. Nandakumar. Please.
Principal, sir, may I ask you to present the certificate to Dr. Biju Matthew?
Hello.
Sir, may we request you to accept our certificate? I now request all the participants, if you have any doubts, kindly post your questions in the chat box so that sir can answer the questions. Sir, I would like to, put a word that, many of them have given a number of, appreciations, and they have given comments like, it was a very clear and wonderful presentation. So thank you very much for that, sir. Sir, now our principal sir is back. I would like to, represent the certificate. Principal sir.
Yes, Nandakumar,
I
please.
request you to hand over the certificate, e-certificate to Dr. Biju Matthew.
Thank you, Dr. Biju Matthew, sir, for nice presentation. So really, this is very basic and essential. So thank you very much for your presentation, patience and presentation.
Thank you very much, sir.
Thank you.
So if there are no further queries, I think it was a very clear concept by our speaker. So there are not much of queries. So with that note, I would like to thank our speaker for giving his valuable time. And now may I request Dr. S. Dhanalakshmi Madam, Associate Professor, Faculty of Pharmacy, to give the vote of thanks. Over to you, ma'am.
Thank you, sir.
Any questions? Any questions or any clarification?
Yes, sir. Now questions are coming up. There was a question from Ashutosh Kumar Singh. Can bond strength affect the intensity of drug action?
Ashutosh Kumar from there.
Yes, sir.
Hello.
Yes, sir. I have a question, sir.
Okay. Okay. Tell me, Ashutosh.
Sir, Mr. Ashutosh has asked, can bond strength affect the intensity of drug action?
Yeah, obviously. So you see here the bond strength and bond energy. So that it will never tells you in a clear picture in molecular docking. If you are going for dynamics, so you will get a clear picture how the strength will be affected. Obviously.
Thank you, sir.
Okay, Ashutosh.
Thank you. I should convey my thanks once again to Dr. Biju Matthew, sir, for his very clear animated presentation. I think the basics of receptor interaction are very clear to all our students and the participants. May I again request Dr. Dhanalakshmi Madam to present your vote of thanks.
Thank you, sir. Good morning, everyone. I am Dr. S. Dhanalakshmi. I am here to present vote of thanks for today's webinar. I would like to thank our today's speaker, Dr. Biju Matthew, for enlightening us with the knowledge. Today's webinar was full of knowledge and interesting things. It gave deep insight into today's topic and also revealed some interesting animations also. Once again, I would like to thank Dr. Biju Matthew. I would like also thank our management, our chairman sir and beloved president sir and respected secretary sir to give the constant support. I thank our VC ma'am for her constant support to the FOP event. I would also thank our principal sir who is a pillar of faculty of pharmacy and also giving all the supports to all the faculty members mainly in the events of FOP. I want to thank Dr. S. Nandakumar sir for making the flyer and Dr. C.N. Hemalata ma'am for the visual platform and the event coordinator Komal ma'am as well as Dr. Anusha ma'am and all the faculty members of FOP and other institution faculty members and today's participants. I would end my speech here. Thank you. Thank you once again, sir. Thank you all.
Thank you, madam. To the kind attention of all the participants, the feedback come e-certificate form is provided in the chat box. I request everyone to kindly fill in the form with the correct email ID to receive the e-certificates. With that note, I thank all the organizers, the speaker and our beloved principal for yet another successful conduction of the webinar. We have more of them coming up, lined up very soon. And I request all the participants to support us in a similar manner. Each time with all your support, we have been very successful in conducting these webinars and we expect and request similar support in the future also. Catch up all of you very soon in yet another program very soon. Thank you one and all. Once again, I request all the participants to fill in the feedback form that is put in the chat box. Kindly fill it up, to get your e-certificates. Thank you very much. Thank you, Dr. Biju Matthew, and thank you all the organizers and all our faculty members and participants. Thank you. Thank you very much.